Chapter 13 is about flow cytometry, and then there's a little towards the end about laboratory automation as well. The essence of flow cytometry is that it's a process that analyzes cells or beads in a fluid suspension and assesses their light scattering characteristics. It often typically uses fluorochromes to identify those cells or particles by their size, shape, and antigenic properties. A fluorochrome is simply a molecule that fluoresces, and by that we mean that if a light of a certain wavelength strikes that molecule, it will emit light of a different wavelength on the way out. And of course, when light changes wavelength, it changes color. Flow cytometry provides rapid and accurate detection of cells found in small numbers. Its uses include to diagnose HIV infection, diagnose leukemia and lymphoma, and accurately categorize them according to what type they are. There are functional assays for various conditions, and we can immunophenotype cells. In other words, we can examine cells, determine exactly what markers they have on their surface. Specimens that can be used for flow cytometry analysis include whole blood, bone marrow, fluid aspirates, and tissue samples. Whole blood is typically collected in EDTA, stored at room temperature for up to 30 hours. If the storage time is thought to or expected to be longer, it may be preferable to use heparin for whole blood, and heparin is also used for bone marrow. If you are in the position of needing to collect blood from a patient for flow cytometry analysis, as always, you would refer to your lab's SOP for that particular test and collect either in EDTA or heparin as specified. When it's time to test the blood, it must be well mixed. If the blood arrives hemolyzed or clotted, it cannot be used for this test, and the specimen must be rejected and recollected. Tissue specimens are going to be collected by the physician. They should be transported in tissue culture medium, store them at room temperature if they're going to be tested immediately, or put in the refrigerator 4 degrees if the testing is delayed. Specimens that contain a lot of red blood cells require that the red blood cells are removed to allow for the white cell analysis. In other words, the red cells are in the way at that point, and we need to get rid of them. Currently, density gradient centrifugation is used to remove those red blood cells. If we're considering tissue specimens, they need to be disaggregated or broken up so that individual cells are present. Some methods of doing this include cutting them up with scalpel and forceps, using a needle and syringe, pushing them through a wire mesh screen to break them up. After this processing is done, then antibodies are added. These are going to be labeled antibodies, most commonly with a fluorescent tag, and these are going to be almost always monoclonal antibodies because we want them very specific for the particular CD marker or other analyte that we're testing for. This simple graphic reiterates the specimen processing aspect of this, whereby specimen is collected from patient according to what the doctor has ordered. The cells are going to be prepared in the appropriate container. They may, if they've come, say, from a tissue specimen, It may need to be broken up appropriately, but the point is we want to end up with individual cells. If there were excess red cells, we would have centrifuged the specimen to get rid of those. And then that cell suspension is treated to fluorescently tagged antibodies. Time is given for those antibodies to combine with the antigen. And then ultimately, we're going to use fluorescence detectors to measure the amount of fluorescence of the various wavelengths that are in the specimen. And then after processing, here's our processed specimen, whatever it might be. We have added those labeled antibodies, allowed time to elapse, and then this mixture of cells that's in a fluid environment is drawn single file through this device, and there'll be an aperture. Once we establish this single file flow of the cells, there'll be an aperture with a light shining through, and as that light goes through, it will detect fluorescence. And in order to detect the different types of fluorescence, we'll have to have different fluorescent molecules that were attached to the original antibodies, and we'll have to have detectors that are detecting fluorescent at the appropriate wavelengths. We will also be assessing the forward light scatter, what light comes through here, and also the light that comes through from the side, and ending up with what's called a dot plot. It's a graph, and the interpretation in the graph involves recognizing the different areas. These are going to show up as dots on your actual graph, but here they're nicely color coded, showing you that each one of the red labeled cells in our example are going to show up in this part of the graph. Our blue labeled cells are going to show up on this part of the graph. There's really four basic components involved in flow cytometry. The first is we have to have fluids, because these cells that we're interested in assaying are suspended in fluid. We need a laser light source, which is going to generate a very narrowly focused band of light that's going to pass through a lens and an aperture to strike cells that are suspended in our fluid. We're going to need the photodetectors to detect signals as they are emitted from this aperture. So we're going to measure the light 180 degrees from the laser, the forward angle. We're going to measure the light as it comes out at right angles, the side scatter. And then we can have detectors that detect only light of certain wavelengths. And as you know, the wavelength of light determines what color that light is. So depending on your instrumentation, you can have a different number of fluorescence detectors. The reason we're measuring fluorescence is that, if you remember, a fluorescent molecule is one that absorbs light at a certain wavelength, but then it emits light of a different wavelength. So it might emit yellow light or green light or what have you. And so those different light colors are measured by different detectors. And then all this data goes straight to the computer. And once all the data is in the computer, we can manipulate it and play with it in different ways in order to supply the information that we're looking for. In the previous slides, you might have noticed that we're going to measure both intrinsic parameters and extrinsic parameters of our cells in solution. Our intrinsic parameters are forward light scatter and side scatter. In other words, where I showed you where the detector is directly opposite the laser, or whether it's to the side. The forward light scatter, you'll see it abbreviated FSC, forward scatter, is an indicator of the size of the cells that are passing through the aperture, while the side scatter, SSC, is an indicator of the granularity, the inside texture, if you will, of the cells, and that helps us determine how complex it is internally, which helps identify it. So the combination of the two can be used to differentiate lymphs from monocytes from neutrophils. The extrinsic properties or parameters of the cells are what we're going to assess using those fluorescent probes. The fluorescent labeled antibodies that are there have bound to a cell with antigens that match those antibodies. And then using those fluorescent molecules with various emission wavelengths allows for the simultaneous evaluation of several properties. It's the combination of data, the FCC, SSC, and the fluorescence that allows for the characteristics of cells according to size, granularity, DNA and RNA content, antigens, total protein, and cell receptors. Once that data has been collected and sent to the computer, we can start manipulating it. So what you're looking at here is a single parameter histogram. The term histogram literally translates a record of cells, single parameter, meaning that we're just looking at one property of those cells. So the y-axis is the number of events. So as we go up the y-axis, the numbers get bigger, indicating that more and more of those were recorded. The x-axis is the parameter to be analyzed, and that's chosen by you, the operator. It is usually an extrinsic parameter, such as a fluorescent labeled antibody. The operator can then set a marker to isolate positive events. We'll look at that in a moment. And then the computer does the hard work for us and calculates the percentage of positive events within the designated markers. Now we're looking at what is termed a dual parameter dot plot. We're looking at a group of cells and we're evaluating two parameters simultaneously. This was whole blood, and we have, before we analyze this thing, we lysed the red cells, typically, because we're not interested in those. That's not what flow cytometry is all about. We're interested in the white cells. And so then after running this specimen through, and this is white cell or red cell remnant pretty much down here, so we don't care about this. But in this case, we've decided we're interested in looking at the neutrophils. And so then we as the operator draw a gate, and all a gate is it's like drawing a circle around something you're interested in. You're marking and you're telling the computer, let's look at what's inside here and expand that and evaluate it, and let's ignore all this other garbage on the outside. Here we're examining a dual parameter dot plot. Now we have chosen which two parameters we want to look at. How did we choose them? Well, we drew a gate around them on the computer screen. So basically each axis, there's positive fluorescence and negative fluorescence. So over here on the y-axis, anything that is up here exhibits positive fluorescence for whatever that blue color is assaying. And down here, that is cells that don't fluoresce at that wavelength. And then over here, we have cells that are positive for the one color and positive for the other as well. So these are the dual positives up here. So if we try to plug this in to an actual chart, we're looking at CD4 and CD3. How are we looking at the two different ones? One, the antibody to CD4 is complex to one fluorochrome. The antibody to CD3 was complex to a different fluorochrome, such that they emit different colored lights. So what we end up with here, the top right-hand corner, these fluoresced both. We had fluorescence in this area for both colors. And so these are CD4 positive T cells, and they also have the marker CD3. The clinical applications for flow cytometry are many, including using particular CD markers for diagnosing and monitoring leukemia, lymphoma. We can use it to count CD4 T cells, becomes very important in the diagnosis and the treatment of HIV. In stem cell transplantation, a good use is to count CD34 cells because these indicate that the cells are indeed stem cells. We can use it to determine the ploidy status of tumor cells. If we're looking for a specific antigen that is found in tumors but not in normal cells, we can use it to identify those, and we can use it to count. We're going to switch gears just a little bit here and talk about lab automation in general. Anytime we can automate a process, by definition, the inherent errors are going to decrease because you take out those occasional human errors that occur. It's going to lead to test results that are more accurate and precise. It's going to require fewer live bodies around the lab in order to perform all these tests. Leads to a much smaller sample size, which has a number of benefits, including saving money because we're using a lesser amount of the controls. If we have to dilute and repeat things, everything is just less, and that's a better thing. Often the modern instruments have barcoding, so that a barcode is on the patient specimen that is automatically read by the computer with the analyzer. Again, eliminates potential human error, speeds the entire process up. Now we can kind of divide analyzers into two categories, and the one category is batch analyzers, where you load these things up, okay? And so I might be able to run 50 specimens at a time, but generally it's going to run all 50 of those before it then goes on to the next assay type. And so we do have access to the samples, which is a good thing, but the negative thing is one type of analysis at a time. Random access analyzers are an improvement over batch analyzers because you can program the instrument to run, these 5 tests on specimen 1, 2 tests on specimen 2, 10 tests on specimen 3, etc. And it will finish up a specimen at a time rather than waiting to finish up a specific test type at a time. These are basically the tasks that the instrument does for us as opposed to what we did manually years ago. So the instrument, once you put the sample into the instrument, it samples or sips the sample, if you will. It adds reagents, mixes them appropriately, incubates for the correct amount of time. The detector's built in, and then the data obtained from the detector goes to the computer. The computer makes all the calculations and spits out a printout and or captures the result to be electronically transferred back to the physician or the patient's chart. The term validation is really a term that includes the measurement and the parameters of a given assay and the assurance that that assay is doing what you think it's doing, what it should be doing, whether it can be trusted. So let's say you're in a lab and a new piece of equipment is purchased to do test XYZ. You don't just have it delivered, open the box, set it up, and run with it. You have to validate that instrument and that methodology for that particular assay by doing some fairly extensive testing so that you can be as confident as possible that the instrument and the method are functioning the way that they should be. So there's some terms associated with this. I think you've seen most of these before, either in this course or in other courses, or you will see them in the future, but it's important to get a good grasp of these, so we'll look at them. Accuracy is defined as the closeness of the value you get with the actual value. So let's say you have a standard. It came in a vial produced by a manufacturer. They assayed it, let's say 1,000 times on equipment that was known to be accurate. And so they're telling you in that bottle, the right answer is such and such. And then you start running that specimen to see what kind of answers you get. So your results are deemed to be accurate when you're getting values that are very close to what the true value is deemed to be. And we always use targets to describe accuracy and precision because they work and most of us can identify with the target. So, if the bullseye is defined as the right value for something, and you're running this specimen many times and you're consistently getting very near to the right value, you can be convinced that your machine is accurate. Precision refers to reproducibility. Are the answers that you're getting tightly grouped? Okay, if they're all over the target, yeah, we got a couple here, one at least on this example, that is accurate. So that's a good thing, but it's not precise. And so therefore, we have an issue. We can't necessarily trust a given answer that comes out of that instrument. And it is entirely possible for a result to be precise but inaccurate. You have a nice tight group, but you're not hitting the bullseye with it. So further evaluation needs to be done. I'm assuming most of you are familiar with firearms. And so, I think about it when you're sighting a gun and you're literally shooting at a target. This is what you want, of course. You want to be accurate and precise. But when you're getting precision but not accuracy, well, you need to adjust the sight, the way you're aiming, or, physically adjust. Well, back the guns I used 100 years ago, you physically, adjusted the sight a little bit in order to take these nice, precise shots, which are good, but bring them so they're accurate. They're hitting the bullseye as well. Moving down to sensitivity and specificity, we've gone through these earlier in the semester. Sensitivity used this way is the lowest measure amount that you can detect. Okay? Specificity is the ability to generate a negative result when the substance you're testing for is not present. So these are defined a little bit differently here, but they absolutely mirror what we learned about sensitivity and specificity earlier. The reportable range is a determination of just how high and how low are the values that you can detect and feel that they're correct. And the reference interval, most of us typically refer to as the normals, the normal range. And this is what somebody determines is that, within this range, this person does not appear to have pathology associated with this analyte. I'm sure in your other courses you spend some time on the determination of reference values or normals and the statistical analysis that allows us to come up with normals. So I won't go into that here other than to remind you that what's the old saying? Normal's a setting on your dryer. Because it's a statistically derived number, it is entirely possible for someone to be a little higher or a little lower than the reference level and still be normal in terms of health. And patients don't always understand that, and so sometimes they'll see a lab result, and if a normal hemoglobin for let's say a normal hemoglobin for a woman is what is it, about 11.5 to 12.5, somewhere in there. And she's, 11.3, and suddenly, oh my God, I'm sick. So sometimes you have to kind of talk your way through these things with patients, family members that get lab results that don't understand. And reference intervals are different for different age groups, sometimes for males and females. And those differences are not always spelled out in a lab report. But certainly the physician knows that if he's testing a 10-year-old for something, the normal range is likely different than if another physician is testing a 70-year-old for the same thing. So if you're not in the know about reference intervals, you will be made aware um, in your career as a student, but always keep in mind where they come from. And they're not magic. They do change over time as we gather more information medically. I think of blood pressures, um, which are constantly being adjusted by the AMA and other medical groups, where a blood pressure that was considered borderline elevated a couple years ago might be considered normal today, and on and on. To summarize, flow cytometry is a method that identifies and counts various cell populations by measuring their properties while suspended in a moving fluid or liquid medium. Each cell or particle passes single file through a laser light source, producing characteristic light patterns that are measured by multiple detectors for scattered light and fluorescent emissions. Scattered light in a forward direction is a measure of cell size, where the side scatter determines a cell's internal complexity or granularity. A single parameter histogram shows a chosen parameter on the x-axis versus the number of events on the y-axis. A dual parameter dot plot displays two parameters plotted against one another. When we're analyzing data, a gate is what we tell the computer to mark off, consider the data inside this area that we've gated off, and evaluate that and ignore the other data for the moment. Flow cytometry is the most commonly used method for immunophenotyping of lymphoid and myeloid populations. It continues to be probably most commonly used to assess CD4 T cell counts. Clinical immunology labs are using a variety of automated immunoassay analyzers. Validation must be done on all assays for all automated systems. Accuracy refers to a test's ability to measure what it actually claims to measure. Precision is the ability to consistently reproduce the same result on a given sample. Analytic sensitivity is the lowest measurable amount of an analyte. Analytic specificity is the assay's ability to generate a negative result when the analyte is absent. The reportable range is the range of values that will generate a positive result for the specimens assayed by that test procedure. The reference interval, or normal, is the value range found in healthy individuals who do not have the condition that is detected by the assay. And always keep in mind, normals vary from method to method, lab to lab, population to population.